Welcome to chapter 3.5. This is the last section of chapter 3. So this is the last video before your Unit 1 exam. So chapter 3.5 is all about advanced microscopy. So things like electron microscopes and fluorescent microscopy. But before we get into chapter 3.5, let's do a review question. Okay, so review question number 3. Hypothesize what you would observe if you forgot the counterstaining step when staining a gram-negative bacterial cell. A, the cells would appear dark pink. B, the cells would appear purple. C, the cells would be colorless and difficult to distinguish. Or D, the cells will wash off the slide. Okay, so take a second, pause the video, and see if you can't find the answer to this question. Okay, so let's go through the answer for this question. So the answer for this question is C. The cells would be colorless and difficult to distinguish. So let's go through the steps of the gram stain. So after we have our sample on the slide and we have it fixed, whether by chemical fixation or heat fixed, okay, the next part is to actually do the gram stain. So the first stain we apply is the primary stain, which is the crystal violet, which is going to stain gram negatives purple as well as gram positives. Okay, so we're going to rinse off the excess stain, we're going to dry it, and then we're going to add the mordant step, or the iodine. Well, that's set for a minute, and what's going to happen when a gram positive, it's going to complex with the iodine and the crystal violet and cause the crystal violet to stick in that thick peptidoglycan layer. But with gram negatives, we don't have that thick peptidoglycan layer. So what's going to happen is the iodine and the crystal violet are going to complex, but it's not good to stick because it doesn't have that thick layer. Okay, and then we're going to rinse off the excess iodine, and then we're going to do the decolorization step. Now, remember, this is the most important step of the gram stain. Okay, so this is what's going to wash away or decolorize the cells that don't have that crystal violet complex, crystal violet iodine complex in its peptidoglycan layer. Okay, so for gram positives, they stay purple after this step, and for gram negatives, they become colorless. Now, the last step of the gram stain is the counter stain step. This is where we add the saffron in and we get the nice pink color gram negatives are going to be. So if everything went according to plan and you followed every step, you'd get a pink gram negative cell. Okay, well, the thing is we forgot the counter staining step. Okay, which means we went through the primary stain, we went through the mordant, and we went through the decolorizer. Okay, so right now, that after the decolorizer, gram negatives are colorless. So this is why this is the answer C, because we didn't come back and do that counter staining step. We haven't added the saffron in to make them pink. Okay, but we've removed the color so they're not purple either. This is a good question to know. Okay, I say it might show up in both the class exam and lab exam. This is not a bite, it will. Okay, so remember, I told you that you needed to know the gram stain inside and out. Okay, this would be an example of the type of question you would see about the gram stain on the test. Okay, that brings us into chapter 3.5, advanced microscopy. Now, this is a really cool chapter if you're interested in microscopes and microscopy. For the class, you will not have any homework on it and you will not be tested on it except for fluorescent microscopy. We need to know a little bit about fluorescent microscopy or for clinical diagnoses and for things like we've already talked about previously in the class. Okay, so this is just a for your information section for the most part. Okay, so what we're going to do is we're going to talk about mostly fluorescent microscopy and why it's important. But if you want to know about different types like electron microscopy, you can read the book. It's actually pretty cool if you like microscopes like I do. Okay, we are at the last case history for chapter 3. Okay, so our last case history is from hot tub to hospital, and it's all about Jared, a 48-year-old man from Columbus, Ohio. So Jared goes to the hospital after 6 days of feeling weak and short of breath. I'm not even going to try to say what that's referred to. Other symptoms he had when he went to the hospital, he had muscle pain, so myalgia. He was nauseous, he was vomiting, and he had pretty terrible diarrhea. Okay, so when he was admitted, he was feverish, he was flushed, and he started coughing with a dry cough. Okay, so with the cough being one of his latest symptoms, the hospitalist group in the ER decided to go and give him a chest x-ray to see what's going on. So his x-ray showed evidence of pneumonia with a mass appearing in the lower left lung. So before Jared started to feel all these symptoms and before he went to the hospital, when he was asked what he'd been doing, he admitted that he had cleaned out the filter for his outside hot tub. Okay, so they took a sample of Jared's sputum. Okay, remember this is the phlegm and the spit, so the snot, and they cultured both his sputum and they took a culture of his spa filter. So what happened is when they grew both of these cultures in the lab, the bacteria were found to be a gram-negative bacilli. So remember, bacilli, those are the rod-shaped bacteria. Okay, so gram-negative, meaning it showed up pink on the slide after we do the gram stain. Okay, so they also ran it through another microscopy test where they used the antibody stain. So they used a specific type of antibody. So they were looking for bacteria that grow well in warm, wet conditions. Okay, so they used the antibody for the bacterium Legionella pneumophila. Okay, and when they ran the test and looked at it underneath the microscope, they saw these little green guys. Okay, so he tested positive from Legionella pneumophila. Okay, so he clearly had pneumonia. The mass in his lower lung was actually the infection. Okay, so Legionella pneumophila, okay, is a bacterium that is found mostly in warm, humid climates. So if it first appeared back in the Legionnaires, like when they were going through Africa, because of warm, wet climates, we call this Legionnaire pneumophila, so the type of pneumonia that causes is called Legionnaire's disease. And we cause the Legionnaire's disease because there was a group of Legionnaires at a conference in a hotel, and the filters in the heater were contaminated with this bacterium, and every one of the people at that Legionnaire's conference actually came down with this type of pneumonia. Okay, so once they figured out what bacteria was causing this pneumonia, Jared was treated with IV amoxicillin and clarithromycin for 8 days and was able to recover fully. So this is a good example of how different types of microscopy tests can help us diagnose different diseases. So we went through two major stains here. We did both the gram stain and we did the antibody stain to figure out what was going on. Okay, so we need to talk about antibodies. Antibodies are immune molecules. Okay, so they're proteins. Okay, and they have a very distinct Y shape. So as we see here, okay, we have this Y. So at this end of the Y, okay, so we call this the tail. This is a fluorophore. This is our glowing molecule. Okay, this is what's going to allow us to see it underneath a fluorescent microscope. Okay, but the thing is with antibodies is they're very specific for something called an antigen. Okay, so this is going to be a protein or any type of molecule that it can attach to. So if we have our pathogen here, and this is the triangle antibody that's on the outside of it, and we want to see if it's actually there, what's going to happen is these arms of the antibody are going to have an area specific that will bind specifically to this triangle and nothing else. So when this bacterium with these antigens are in your bloodstream, your antibodies are going to go and bind to them, and in your body it's going to signal your immune system. For microscopy, we're going to look at this underneath the fluorescent microscope and see glowing bacteria because it was found in the sample. Okay, antibodies are very specific, which is why we use them for the antibody stain. So when we're looking for specific pathogens, and we kind of have an idea that we might have that pathogen, this is one of the tests that we use. Okay, so fluorescent microscopy is a very powerful clinical diagnostic tool because of the antibodies that we use, because they're so specific that if we see evidence of fluorescence in a sample, that means that that antibody had to bind to that specific antigen, meaning that specific pathogen was in our sample. Okay, it's also pretty powerful because it detects specific, not just because it detects specific microbes and sometimes different subcellular structures like DNA, but it also will remove the background noise. Okay, so we won't get any signals for things like dust or non-specific materials. So they're kind of excluded from the microscope slide and from what we're viewing with the fluorescent microscope. Okay, so we use the fluorophore or the glowing molecule to highlight certain things. We use fluorescent microscopy a lot to count living versus dead cells. So we have a saying in microbiology, green means alive, red means dead. Okay, so in this slide, you can see we have the living cells, the living bacterium, these are going to be the green, and then we have the dead bacteria, which are going to be the red. Okay, so sometimes with fluorescent microscopy, we'll use actually different antibodies all in one sample, so we can highlight different things. So in here, we'd actually have two antibodies, one specific for the living bacteria and one for the dead bacteria. Okay, because dead bacteria are going to present different things on the outside of their cells compared to the living. Or we can use it to image tissue samples. Okay, so this is something that we've observed before in class. We've seen this micrograph. Okay, so we have three different fluorescent stains here. Okay, we have one that's going to highlight the villi. So this is a gut cell. Okay, so we're looking for Vibrio cholerae, which is a bacterium that causes cholera. Okay, so we have one fluorescent antibody that's highlighting the villi. We have one that's binding to the mucus, or specifically the mucin protein in your mucus. So that's going to be one color. And then we have a third antibody that's specific for the bacterium, Vibrio cholerae, and it's going to highlight that as green. So we can see the difference between the tissue, the mucus, and our infection, our bacterium. Okay, so this is one of the reasons why fluorescent microscopy is such a powerful diagnostic tool. It's because we can actually image more than one thing at once. And sometimes when we are running the samples in the lab, what's going to happen is we're actually going to make a cocktail, and we'll have different antibodies that glow different colors, and then we'll look for specific toxins. So if we see one color, we know we have this toxin. If we see this other color, we know we have this other toxin. If we don't see anything, we don't have any of them. Okay, and same with the antibodies that are specific for bacteria, because sometimes a fluorescent antibody is for a toxin is not going to be there if we see the bacteria, and vice versa. We might see the toxin but not the bacteria. We might see the bacteria but not the toxin. Okay, or we could see both. Okay, so and we have tests that will show for a lot of these bacteria that produce toxins, we actually have antibodies specific to both the toxin and the bacteria. So it helps us to figure out what's going on. Okay, so one of the last things we're going to talk about is viruses in microscopy. Viruses are really tiny. If you remember from that previous slide, the herpes virus was 0.2 nanometers, or 0.2 micrometers, I'm sorry. So it's a very, very small particle. Okay, we do not have the resolution to see those underneath a light microscope. In fact, the only time we can visualize viruses and toxins, to be honest, is using electron microscopy and using an electron microscope. We have to have that higher resolution in order to be able to see them and magnify the sample enough to see something that tiny. Okay, so we have two micrographs here from electron microscopes. We have this is actually the HIV virus. Okay, so these HIV particles, we call them virions. Okay, so this is a T cell. This is what the HIV virus infects. This is what it targets in our bodies. And so we can see that all these little viruses that were made are actually budding off of this T cell and being released into the bloodstream to actually go infect other T cells. So that's kind of cool. Now, something that we're a little bit more familiar with would be the SARS-CoV-2 virus or the COVID-19 virus. So this is a electron micrograph of COVID. So we can see on the outside, so we have the virus particle here, the virion, and we can see these really interesting looking spike proteins on the outside of it. Okay, this is actually what our immune system, so if you get the COVID vaccine, you're going to have, it's an mRNA vaccine, and it's going to make a protein that looks just like the spike protein. So it's going to train your immune system to look for the spike protein. Okay, so after you get the vaccine, if you catch COVID, what's going to happen is your immune system's going to be like, oh, I recognize this guy. This is not part of us. Let's target it and we need to get rid of it. So this is the actual particle that our vaccine trains our body to look for. Okay, so that is chapter 3.5. We have one more review question we're going to go through, but this review question requires us to look at or remember our case study from the beginning of this chapter, the pathogen in a can. Okay, so we have Mariana and Julio. They're two kids from Oklahoma. They eat the commercially canned hot dog chili sauce and get sick with food poisoning. Okay, so they go through all those different symptoms. They have blurry vision, they have the symmetrical weakness, but normal vital signs, and they're starting to have their pupils of their eyes are going into paralysis. Okay, so they're fixed, so they're not adjusting to light. Okay, so the paralysis moves slowly from their eyes and from their face downward and starts to hit their lungs. Their breathing starts to become difficult. Remember, they got rushed to the ER. They had botulism because the botulism toxin was found in their system. Okay, so you can read the rest of the slide and pause this before going on to review question number 4 if you'd like. But let's go to the review question. So the review question says, think back to the case study from the start of the chapter. See next slide if you don't remember. We actually put this previously in your notes. It's going to be the next slide. But it was previous here in the video, so you could read it before the question. So why was fluorescent microscopy used to help identify the causative agent of the children's disease rather than bright field? Okay, so think about what we learned about microscopes, think about what we know about viruses and toxins and bacteria, and try to come up with the answer about why we chose fluorescent microscopy other than our light microscopes. So pause the video and see what you come up with. Okay, so let's talk about this. So one of the things that the test for both Julio and Mariana showed is that they had presence of the toxin, but they didn't have presence of the bacterium in their bloodstream. Okay, so if we had done a gram stain of their blood sample and we're looking for the bacterium, we actually wouldn't find it. Okay, because we only found the toxin. Now, toxins are tiny proteins. Antibodies are tiny proteins. Okay, we wouldn't be able to see the toxin in a light microscope. Okay, and since there's no bacteria, when we looked at the sample underneath the light microscope, we saw nothing. Okay, so one of the key things about the fluorescent microscopy is that we use those antibodies that are specific to certain molecules, so they're very specific. So we looked specifically for that toxin. Okay, and by using that antibody and fluorescent microscopy, we were actually able to see it without having to go to an electron microscope, because electron microscopes are not a common thing that you see at hospitals. Some hospitals do have them, but most do not. Okay, so being able to specifically select for that toxin using the antibody and then seeing that pop up underneath the microscope was an easier way to diagnose what the children had than, say, waiting for a micrograph of their blood to come back from an electron microscope. Okay, but pretty much the reason why we used it is because we wouldn't be able to see the toxin in using a light micrograph. Okay, so that is all of chapter 3. We are done. Congratulations. We are now ready to take the first exam. So we'll see you in class.